How do we know a health intervention is safe and effective? This is basically the reason for clinical trials. Over time, it became evident through various explorations, some accidental, that an intervention such as, let's say, a pharmacological agent, a drug, could influence a disease. For example, penicillin was first found to kill bacteria, many years ago. And so when you take this substance and you say, well, can this influence a disease? Can taking a cholesterol-lowering medicine, for example, reduce the risk of heart attack or stroke? To find out, we have to engage in clinical trials, and these work at different levels. They begin with what we might call safety studies. Give a group of patients this experimental medicine and find out if there are any problems, side effects, or issues there. Assuming it clears that hurdle, then you can move up to the larger trials in which you have a group of patients with a disease or risk of disease, and you randomize them into groups. One group gets the experimental treatment, one group gets a placebo. The patients and the researchers are blind to who's getting what, and the data is collected over time. And in those trials is when we discover, is there a statistical significantly different degree of disease or reduction of disease in those two groups. And that's basically how we determine if interventions are safe and effective.